So this presentation is on a recent study by Sergei Kouzov and co-authors at St. Petersburg State University, comparing the diets of 3 swan species at spring staging sites in the eastern part of the Gulf of Finland in the 21st century. The diet of swan species has been studied by researchers in different parts of the globe over many years, with the greatest number of publications relating to the mute swan and the tundra swan. Most of the data on mute swan feeding has been obtained at inland water bodies in Western Europe, and the data on tundra swan feeding refers mainly to food taken in the wintering range. No studies of swans' diets have been conducted in the Eastern Baltic and in the Gulf of Finland until recent times, despite the region being of a major importance for its staging sites used by all 3 of the swan species migrating along the Northwest European flyway, which are located here. Both Hooper and Buick swans accumulate energy reserves on the Gulf for their onward flight to the next staging site on the delta of the northern Dvina River in the White Sea region and for breeding. Mute swans also need to accumulate energy reserves by feeding in the Gulf in preparation for breeding on nearby islands. The objectives of the study were, number 1, studying the feeding resources of swans at migratory sites. Number 2, exploring differences in the diets of the 3 species when sharing habitat, using the analysis of fecal samples. And lastly, explaining the identified dietary patterns based on the morphology and history of the species. The area with the most numerous migration stopovers of swans in the southern sector of the Gulf of Finland, the Kurgalski Peninsula, was selected for research. Fecal samples were collected between 2014 and 2019, and in these years, spring came early. In particular, the dates of ice-free waters were very early. In these years, migrating aggregations of swans appeared very early and were the most abundant. Mute and Hooper swans usually appeared in February. The peak of the number of mute swans was noted in the first half of March and of Hooper swans in the second half of March. Buick swans appeared at stops during the second half of March, with maximum numbers observed during the second and third parts of April. The last Hooper and Buick swans were seen until mid-May. Mute swans that did not breed formed pre-breeding aggregations and continued to stay in the study area in high numbers during May and the first half of June. We collected 233 fecal samples over 6 years, 87 samples of these mute swans, 77 Buicks, and 69 Hooper. We divided each spring season into 3 periods, the first period from 1st of February to 15th of March, the second period from 16th of March to the 30th of April, and the third period from the 1st of May to the 15th of June. The greatest number of samples of all 3 species was collected in the second period, and this was 158 samples in total. During the first period, 35 samples of 2 species, mute swans and Hooper swans, were collected. In the third period, we collected 40 samples of only 1 species, the mute swan. A total of 44 plant species were detected in samples of all swans, 24 species of macroalgae and 20 species of vascular plants. The greatest species diversity was observed for green algae and submerged vascular plants, 10 species in each group. In mute swan samples, the greatest number of macroalgae species was observed, 24 species. In Hooper and Buick swan samples, the greatest diversity was observed in vascular plants, 17 and 19 species respectively. In February and the first half of March, only mute and Hooper swans were present at the sites. New aquatic vegetation had not yet begun to grow. The main food resources at this time were scraps of last year's cladophora and pondweed branches that had overwintered. As can be seen from the table, the diets of mute and Hooper swans during the February and the first half of March are very similar. Cladophora green algae dominate, and cladophora clumps are often present in overwintered branches of pondweed, and swans eat them all together. The essential amount of detritus in the diets of both species, in our opinion, is also caused by eating it in clumps of cladophora. In the second half of March and in April, all three species of swans were present at the site, and Buick swans dominated. At this time, the vegetation of macroalgae began, young sprouts of vascular submerged, semi-submerged and coastal plants appeared, and the development of their root system began. The species composition in samples of different swan species in March to April varies greatly. Green algae continues to dominate in the mute swan's diet, but diatom algae and vegetative parts of soft submerged vascular vegetation appear in significant proportions. In the Buick swan diet, sprouts of vascular plants predominate. Green algae are in the second place among foods. Thin roots of vascular plants, mainly reeds and pondweeds, also make up an essential part. Thick rhizomes of reed occupy the largest part in the diet of hooper swans. Vegetative part of vascular plants are on the second place among other foods. Algae in samples of hooper swans are much less common than in other species. Diatom algae were found only in fecal samples of the mute swans. The reasons for this are not completely clear. We also did not observe Potamogeton nodules in the mute swan samples. We attribute this not only to the difficulty of determining nodule residuals in fecal samples, but primarily to the low density of this plant in swan sites, as well as to the greater need of birds to obtain protein rather than carbohydrate nutrition in the pre-breeding period. Typical remnants of the meals of the hooper swan and Buick swan in April. On the left are remnants of young reed sprouts at the feeding site of Buick swans. On the right are remnants of thick rhizomes at the feeding site of hooper swans. Multivariate analysis of variance was used to determine differences in the sites of three species of swans. To compare the diet of the three species, only samples collected from the 16th of March to the 30th of April were used, since only during this period were all three species present at the same time. MANOVA was used to evaluate variation in 7 food categories consumed by the swans. They are green alga, brown algae, red algae, diatoms, thin root papillae, and rhizomes and tubers of semi-submerged and coastal plants. Vegetative parts of submerged aquatic or coastal plant and detritus. The content of these categories of food in fecal samples differed significantly. Additionally, we divided all feeds according to the degree of roughness into the following categories: macroalgae, thin root papillae, semi-aquatic grasses, young shoots of semi-submerged vegetation, and Phragmites rhizomes. We found significant differences in the degree of feed roughness among the three swan species. Mute swans consume the softest food of group 1 and 2, algae and soft submerged vegetation. The hooper swan is the only species that widely uses the coarsest food, reed rhizomes. The Buick swan has the widest range of food from algae to young vegetative parts of the reeds. More often than other species, it uses vegetation of the third group, which is coastal grasses and sedges. Differences in swan diets can be explained by differences in beak morphology of the three species. In the beak morphology of the mute swan, filtrating features are most pronounced. This is indicated by the shape of the beak, the largest number of filter plates, the smallest sizes of the maxilla and the mandible nails, the highest density of tactile structures at the end sections of the beak, etc. In the morphology of the beak of the Buicks and the hoopers, on the contrary, nibbling features are expressed, bringing them closer to the structure of the beaks of geese. The beak of the hooper swan has the greatest size and power. The differences in beak morphology and feeding preferences of swans, in our opinion, are explained by the evolutionary history of these species. The mute swan as a species was formed during the warm Miocene-Pliocene in the southern Palearctic, where at that time there was a vast ocean, Parathopus, sorry if that's not right, with numerous shallow eutrophic bays and lagoons with abundant submerged vascular vegetation and macroalgae. The hooper and Buick swans were formed under the harsher climate of the Pleistocene, where oligotrophic, periglacial, and thermokarst lakes of mammoth steppe and dystrophic lakes in the middle of sphagnum moss prevailed. In these water bodies, submerged vegetation, including macroalgae, was less developed, while the coastal zone of these lakes was home to cereal and sedges.